If you've already watched my video on nucleotides, you should have a solid foundation as to what purines and pyrimidines are. If not, I highly recommend checking that video out first. It's short and straight to the point, and it'll also make this video on pyrimidine synthesis way easier to follow. Let's get started. Pyrimidine synthesis takes place in the cytoplasm of all cells, and it begins with carbon dioxide and glutamine, which are converted to carbamoyl phosphate by the rate-limiting enzyme, carbamoyl phosphate synthetase 2, or CPS2 for short. Unlike CPS1, which we covered in the urea cycle video and is found in the mitochondria, CPS2 is specific to pyrimidine synthesis, and it's found in the cytoplasm. To remember this, just think of cytoplasm instead of cytoplasm. This reaction catalyzed by CPS2 is energy dependent, meaning that it requires ATP. When the high-energy phosphate bonds of ATP get hydrolyzed, we get ADP and an inorganic phosphate in return. Additionally, glutamate is released as a byproduct. Carbamoyl phosphate then undergoes a series of 3 reactions, starting with aspartate joining in the first reaction to form carbamoyl aspartate, which then becomes dihydroorotate in the second step, and finally in the third, dihydroorotate turns into orotic acid. The intermediates I mentioned before orotic acid are lower yield, except for maybe dihydroorotate, so I've left them as dots rather than saying their full name. The key takeaway though is that aspartate is used in the first step and that orotic acid is formed in the last. Orotic acid is then converted to uridine monophosphate, or UMP for short, by the enzyme called UMP synthase. UMP synthase works in 2 very high yield steps that are represented by the 2 arrows. First, orotate phosphoribosyltransferase transferase does whatever it says it's going to do, meaning that it transferases or transfers the phosphoribosyl group from phosphoribosyl pyrophosphate, and it attaches that group onto orotic acid to form OMP. Then, OMP decarboxylase kicks in, and as the name suggests, it's going to remove a carboxyl group from OMP to form UMP, which is actually the first pyrimidine nucleotide that's formed in this pathway. Here I've illustrated how the phosphoribosyl pyrophosphate comes in at the first step. It's very high yield to know that this intermediate is also found as an intermediate in the purine synthesis pathway, and thus it can go through either of the 2 pathways depending on the needs of the body. Then, because the second step is a decarboxylation reaction, carbon dioxide is released, which is a hallmark feature of all decarboxylation reactions. Moving on, UMP is converted to UDP, which can then follow one of 2 pathways. It can either be converted to DUDP by the enzyme ribonucleotide reductase, or UDP can be converted to CTP by CTP synthase. Once DUDP is formed, it can then be converted to DUMP, which finally gets converted to DTMP by thymidylate synthase. This enzyme requires methylene tetrahydrofolate as a cofactor. Its function is to donate a methyl group. After losing its methyl group, it turns into dihydrofolate, which is then reduced back to tetrahydrofolate by the enzyme called dihydrofolate reductase. Then, tetrahydrofolate gets remethylated to begin this entire mini cycle once again. This role of folate as a methyl donor and in 1 carbon transfer reactions is not only crucial for pyrimidine synthesis, but also for many other biochemical pathways in the body. This makes this a very high yield concept to understand. Now that we've covered the whole pathway, you're probably wondering, how do I memorize all of these random letters and words that look like someone just smashed their keyboard and called it pyrimidine synthesis? But I have a mnemonic for you that will help you keep this entire pathway right on your fingertips. The mnemonic goes like, pyrimidines create great compounds and drive erotic pathways, ultimately making us develop. The letters in red correspond to the order of intermediates in the pathway. Guys, this mnemonic is so perfect and it flows so nicely. And if you need, you can even pause the video here to match each step with the mnemonic. It will make everything easier to follow. Now, for the last three nucleotides that are not included in the mnemonic, specifically DUDP, DUMP, and DTMP, to memorize them, just recite them like a rhythm or a song. These three nucleotides all start with a D and sound similar, which makes them very easy to remember when you say them all together. Now you're probably wondering, okay, well, I can memorize the intermediates with the help of the mnemonic, but what about the enzyme names? The good news, though, is that they are just as easy to remember as long as you know the intermediates. For instance, the enzyme forming carbamoyl phosphate is literally just called carbamoyl phosphate synthetase, while the enzyme forming UMP is called UMP synthase. The enzymes are named according to what they do, and they usually have either the reactant or the product in the name itself. So if you're able to memorize this mnemonic, and hence the intermediates, you basically have the entire pathway memorized. Now the test writers will actually go further to test your knowledge. And the way they do this is by asking about drugs that inhibit key enzymes in this pathway. Starting off with leflunomide. This drug inhibits dihydroorotate dehydrogenase, which is the enzyme that converts dihydroorotate to orotic acid. Next, we have hydroxyurea, which is an antimetabolite that inhibits ribonucleotide reductase. An easy way to remember this is to pronounce it as hydroxyurea, emphasizing the double R for ribonucleotide reductase. The drugs that inhibit thymidylate synthase include flucytosine and capecitabine, which are both prodrug forms for 5-fluorouracil. Finally, the drugs that inhibit dihydrofolate reductase include methotrexate, trimethoprim, and pyrimethamine, which act on eukaryotes, prokaryotes, and protozoan cells, respectively. Because all three of these drugs have meth in their name, an easy way to remember them is to just say doctors don't do meth. So these are all the relevant drugs that you need to know for pyrimidine synthesis. But what about the diseases? One very high yield condition to know about is when UMP synthase is defective. This condition is known as primary orotic aciduria. The word primary indicates that it is due to a defect in the pyrimidine synthesis pathway itself. This is a genetic disorder inherited in an autosomal recessive pattern. In contrast, secondary orotic aciduria arises secondary to a defect in the urea cycle, most commonly a deficiency of ornithine transcarbamylase. This leads to a buildup of carbamoyl phosphate in the mitochondria, since it can no longer go through the defective urea cycle. Instead, the excess carbamoyl phosphate spills into the cytoplasm and is shunted towards pyrimidine synthesis, leading to the overproduction of orotic acid. Because DNA and RNA synthesis is completely normal in secondary orotic aciduria, we actually don't see the classic symptoms of orotic aciduria. Instead, the orotic acid in the urine is basically just an incidental lab finding, with the main symptoms actually being due to hyperammonemia, which include neurological disturbances and other systemic effects of a defective urea cycle. Now that you know that secondary orotic aciduria doesn't cause symptoms related to orotic acid buildup, you might be wondering, how does primary orotic aciduria, which does present with these symptoms, actually manifest? Let's discuss. Because primary orotic aciduria impairs DNA and RNA synthesis, rapidly dividing cells will be affected, leading to failure to thrive and growth retardation. The buildup of orotic acid in the urine will lead to precipitation of orotic acid crystals, which may cause dysuria as well as other urinary symptoms. Now, for the most highest yield finding on the slide, we'll also see megaloblastic anemia. This is due to impaired DNA synthesis, which leaves red blood cells to be stuck in the immature and oversized state, hence the term megaloblastic. Also very high yield is the fact that this anemia is refractory to folate and B12, because this isn't actually a cofactor deficiency, but rather it's an enzyme deficiency, and thus no amount of folate or B12 could resolve this enzymatic defect. However, one treatment that is actually really helpful is UMP. This is because it bypasses the defective step, UMP synthase. An easy way to remember many of the features listed here is to just think of U, standing for urinary orotic acid crystals, UMP synthase as the defective enzyme, and finally, uridine, or UMP, for the treatment. To remember the highest yield finding of this disease, megaloblastic anemia, just say erotic aciduria instead of orotic aciduria. This connection lies in the imagery. Just as erotic tissue swells up, so do the megaloblastic red blood cells in erotic aciduria. This is a very simple trick, but I promise that you will never forget this if you can remember this key and simple association. And with that, we can wrap up our discussion on pyrimidine synthesis. The highest yield takeaways from this video are the associated drugs, as well as erotic aciduria. I hope that this video was helpful for you, and if it was, make sure to like and subscribe, as well as share with someone else who can benefit from this. Thank you so much for watching, and I'll see you in the next one.